Hello, this is Team Green's presentation on Article 3. Translation termination is a critical process in protein synthesis. Ribosomes stop translation at the stop codon and releases the polypeptide. The key players in this process are ERF1, eukaryotic release factor 1, which decodes the stop codon and cleaves the peptidyl tRNA bond, and ERF3, eukaryotic release factor 3. ERF3 enhances ERF1's function through GTP hydrolysis. A better understanding of translation termination has huge clinical implications, particularly for treating diseases caused by premature stop codons, such as cystic fibrosis and muscular dystrophy. Diseases that are caused by the premature stop codons accounts for 11% of all human diseases. Previous studies have shown that ERF1 and ERF3 are essential for translation termination, but the exact timing and sequence of events remain unclear. Traditional bulk assays could not capture the dynamics of release factor binding in real time, leaving many questions unanswered. However, recent advancements in single molecule techniques such as FRET, fluorescence resonance, energy transfer, and fluorescence spectroscopy have provided a new way to study these events in real time. These innovations have helped scientists track ribosome dynamics during translation termination, paving the way for a deeper understanding of this process. The hypothesis is two eukaryotic release factors, ERF1 and ERF3, in GTP form a ternary complex to rapidly recognize stop codons and trigger translation termination through a multi-step process similar to translation elongation. This process is regulated by UTR sequences and small molecules.
For the first method, in order to dissect the precise sequence of molecular events during termination, the researchers created a highly controlled in vitro yeast translation system. This allowed for exact assembly of ADS initiation complexes using purified components and synthetic mRNAs such as mSTOP and mFSTOP constructs. By tracking peptide release over time, and fitting kinetic curves shown in figures S1A and S1B, they validated that the system could reproduce physiological termination behavior and reliably quantify reaction rates. To observe molecular dynamics in real time, the release factors ERF1 and ERF3 were labeled with fluorescent dyes, which we identified as our second method. This was done using a YBBR tag and SFP enzyme to attach CY3 and CY5 dyes without disrupting function. After purification shown in S1C and S1D, the team confirmed that the labeled factors could still effectively catalyze peptide release, shown in figures S1E and S1F. Ensuring their fluorescence assays reflected true biological activity, this validation was crucial to interpreting subsequent FRET and ZMW results. For method 3, single molecule FRET was used to measure changes in proximity between ERF1 and the ribosome in real time. This technique enabled precise tracking of the moment ERF1 engages the active site and undergoes structural rearrangement. As shown in figure S2C, FRET signals appear when ERF1 bands near the ribosome's 60S unit. Without ERF3, binding was significantly slower and weaker, shown in figure S2D, demonstrating ERF3's role in enhancing binding kinetics and proper positioning of ERF1. These observations provided direct mechanistic insight into the early steps of termination. To observe the arrival and departure of individual release factors with millisecond resolution, zero-mode waveguides were employed as the fourth method. These nanostructures allow fluorescence detection in small excitation volumes, which is ideal for tracking fast molecular events. In figure S4A, simultaneous arrival of ERF1 and ERF3 at the ribosome can be seen, indicating that they often function as a preformed ternary complex. S4b shows a probability distribution confirming consistent binding dynamics across experimental replicates. This setup was essential to resolve the temporal sequence of ERF1 and ERF3 action.
So, since we believe the focus of the article to be on the interactions of the release factors as well as their interactions with GTP, focusing on the experiment depicted in Figure 4 provides the best information to support how they interact. So, this experiment tracks the simultaneous binding of ERF1 and 3 at the stalled ribosome at the stop codon. The initial binding and delivery of ERF1 to the ribosome at the stop codon is arguably the most critical step in translation termination because it directly precedes and is absolutely essential for the subsequent hydrolysis of the peptidyl tRNA bond. So without the correct positioning of the ERF1 at the ribosomal A site, the reaction required for the peptide release can't be properly initiated. So this experiment employed zero-mode waveguides to observe individual ribosome complexes halted at the stop codons. So the researchers then fluorescently labeled ERF1 with Psi5 here shown in red, and the ERF3 with Psi 3.5 shown in yellow, and then lastly they labeled the ribosome with Psi 3, which is shown here in green. So this allowed for real-time simultaneous monitoring of the binding dynamics of both ERF1 and 3 to the same ribosome. So the key finding of this experiment was the frequent observation of the simultaneous binding of ERF1 and 3 to the ribosome. The likelihood of the two independent binding events occurring at precisely the same time over and over again on the same ribosome is very low statistically. So you can see that here shown in Figure B, how they all bound together at the same time. So this observation strongly suggests that ERF1 and ERF3 form a complex before or at the time of their interaction with the ribosome. Furthermore, the researchers found that the presence of GTP was crucial for the co-binding of these release factors. When GTP was omitted from the reaction mixture, the number of simultaneous binding events was significantly reduced, as you can see here in the supplemental Figure 4. This highlights the essential role of GTP in either the formation or the stability of the ERF1 and 3 complex, or facilitating its interaction with the ribosome. But regardless, it's still crucial. Another significant observation was the difference in time spent on the ribosome by both ERF1 and ERF3. The data shows that ERF3 binding was transient and it typically dissociated from the ribosome relatively quickly. As you can see here in Figure C, its time spent bound to the ribosome was pretty low, especially when compared to ERF1 shown here with its time spent at the ribosome much longer. So this suggests that the model, this suggests a model where ERF3 acts as kind of like a chaperone or a delivery factor for ERF1 and just facilitates its initial binding to the ribosome and then quickly dissociates after its GTPase activity. So that allows ERF1 to proceed with the subsequent steps of cleaving the peptide. So this GTPase activity of ERF3 was actually confirmed by comparing the different release kinetics of ERF3 in the presence of different GTPs and GTP analogs. So you can see here in Figure E, we have the rate of the ERF3 time spent with the rapidly hydrolyzable GTP, as well as a slower hydrolyzing analog, the GTP gamma S, and then a GTPase deficient mutant of the ERF3. And from this, the researchers found that the GTP hydrolysis by ERF3 accelerates its release from the ribosome, specifically when ERF1 is also bound. This indicates that while GTP binding is important for the initial interaction, GTP hydrolysis provides the energy that's necessary for the ERF3 to dissociate after it's delivered the ERF1 to the ribosome. So the findings in Figure 4 significantly advance our understanding of how this delivery is achieved. It reveals that ERF3 is not merely an activator of ERF1, but rather an active participant in its recruitment to the ribosome. And this initial binding event, which is orchestrated by the ERF3 and regulated by the GTP, represents a key control point in the termination pathway. Collectively, these findings provide compelling evidence for a model where ERF3 actively delivers ERF1 to the ribosome in a GTP-dependent manner. The observation of the transient ternary complex and the role of GTP hydrolysis in the subsequent release of ERF3 are crucial for understanding the initial stages of eukaryotic termination.
Figure 6 captures the overall mechanism and kinetics associated with eukaryotic translation termination, directly supporting our hypothesis. Panel D is a concise step-by-step model with kinetic timing. Although GTP is not explicitly depicted, it is implied based on previous information from the experiment that it refers to the rapid binding of the ERF1, ERF3 GTP complex to the ribosome, but is stopped at a stop codon, taking just less than a second. Then ERF3 is released once it hydrolyzes GTP, allowing ERF1 to adopt an active conformation and rapidly cleave the peptidyl tRNA bond. The ribosome then quickly rotates, and ERF1 and the peptide are released in about a half a second. This schematic illustrates just how quickly release factor binding is, as it is faster than the subsequent events. In all, termination takes approximately 4 seconds. As release factors are widely conserved from use to humans, it is assumed that this mechanism is fundamental to all eukaryotes. Furthermore, panels A through C support the interdependence of each step of termination, and also illustrate how termination is a multi-step process that is tightly regulated. On the y-axis of these panels are the lengths of the pre-termination and post-termination states of the ribosome. The pre-termination state refers to the point before the peptidyl tRNA bond is hydrolyzed and is characterized by a high Fret signal between the P site tRNA and ERF1 bound at the A site once it is recognized as stop codon. The post-termination state is characterized by a lower Fret signal between the P site tRNA and ERF1 and is the point at which the ERF1 has cleaved and the polypeptide is released. For panel A, different types of 3' UTR mRNA sequences varying in strength from none to strong are shown on the x-axis. These have been previously shown to promote stop codon read-through, which is a phenomenon where a ribosome fails to recognize a stop codon and continues the process of translation. Insertion of the strongest sequence doubled the pre-termination state, but did not affect ERF1 release, suggesting this insertion prevents the peptidyl tRNA bond cleavage by ERF1 and effectively the rest of translation. On the other hand, small molecules like G418 and perimomycin were shown to slow both termination and ERF1 release. Specifically, the addition of 1 millimolar of G418 ultimately lengthened the pre- and post-termination state duration by threefold, while the 50 micromolar of perimomycin doubled the pre-termination state and lengthened the post-termination state by fivefold. As these last few molecules also impacted later stages, such as ERF1 release, by hindering ribosomal rotation, it is evident that all these steps are connected. Considering that many human diseases are due to premature stop codons, which shorten polypeptide sequences, this information could help develop therapies targeting this issue.
This study showed that certain mRNA contexts and drugs, like G418, can slow down translation termination and promote read-through of stop codons. This opens up an exciting possibility. Could we intentionally tune the timing of termination to treat genetic diseases caused by premature stop codons? By slowing release factor activity or altering mRNA sequences, we might increase the chances of inserting a near cognate amino acid, allowing full-length protein production. Future research could explore how to control this process precisely, potentially leading to new therapeutic strategies for diseases like cystic fibrosis or muscular dystrophy.